That's all SciStarter related things. As a database, we support specific or all types of projects. And so today we invited Marianne to talk about the Monkey Health Explorer, which is also featured on the education page on SciStarter. So I'm really excited to learn more, and I'll actually go ahead and drop my screen share so that she can introduce herself. Excellent.
Hello, nice to be here. I'm excited to be able to share about our Monkey Health Explorer project. A little bit about myself. You saw in the little snippet, I am a geneticist. I did not know about monkeys in depth and about their blood, which is what we study, until we were working on this project, and I have learned so much. It's been a lot of fun, so I'm excited to get to share that.
All right, you should be seeing my screen. All right,
Yep.
go ahead.
Okay, so I'll get you started on this project. The name is Monkey Health Explorer because we are studying these rhesus macaque monkeys, like the picture up there on the top left. But first off, I want to share with you the team that is involved in this project, because it's a lot of people involved, and we're looking at many things about those monkeys. But here you can see the top row of the team is my kind of local team, the ones here at the museum. So I work at the North Carolina Museum of Natural Sciences in a research lab that has a glass wall. So we are part of the exhibit. We actually are essentially on display. And we also have a student, Patrick Jacobs, who's from North Carolina Central University, working with our local group here. Then we have on the next row some of our other collaborators reaching out. See, we cross many different organizations. And then the picture in the bottom is just recently, a couple, like earlier in the year, where we all got together to talk about, these are researchers and just everyone who works on this project in some aspect, where we got together to talk about our research, talk about what we're doing, and see how we can work together. Plus, part of our team is our more than 15,000 Zooniverse users who have helped us with our project so far, because our project is hosted on the Zooniverse website, which I'll show you. So we can go on to the next page. So first, a little bit more about the broader part of this research. So this is studying a group of rhesus macaque monkeys that actually live on an island off the coast of Puerto Rico. So you see the map of Puerto Rico here, and then the offset blowing up the southeast corner. So you can see the island of Cayo Santiago, a little bitty island off the side. This is where the monkeys are. They were brought there in the 1930s by researchers to study their behavior. So they're free roaming across the island. They're very social animals. They form social groups across the island. And so they've been studied for many decades now. And more recently, in addition to studying their behavior, they once a year will capture some of the monkeys to take some samples. So we take their blood. Well, I don't personally go down there and do that, but our helpers do. They take the blood and take other measurements. They swab for their skin microbes and many other bits of information. And that's what all these different individuals, like the big crowd I showed you, they're all studying different aspects of these monkeys. The piece that we're particularly interested in is the blood. Because it's just like when we go to the doctor and get our blood drawn, we're taking a sample of the blood and looking at it to get a snapshot of their health. We can also look at the genetics of the monkeys by looking again at the blood, taking the DNA from there and analyzing that. And so I've got our first question. So if you want to go to the next. The question is, can you guess how much of our DNA we share with rhesus macaques?
I like how specific the numbers are. Good choices of it. It'll be in the poll. There we go. See, I want to be able to answer. Ooh, we got some. Well, this is the one I remember the answer to. The other ones I'll be surprised.
And while we're waiting, you can see in this picture, this is some of the monkeys on our island. See how they groom each other. They're very social, and that's a lot of the behaviors that are being measured, is who's grooming who's interacting with who's fighting with who. That's some of the interactions that are looked at. A cute little baby there too.
Yeah, very cute. I'm gonna go ahead and have us stop the poll. Oh, it's sharing right now. We're split, so which one is correct? 93 or 98?
So you can go on to the next slide. And 93%. So that is how much we have in common with rhesus macaques, which is why one of the reasons why we like to study these monkeys, because they're so similar to us genetically. We can actually learn a lot about ourselves by studying them. So we can move back to the next slide, which takes us back to the map. And so on this, like I mentioned, so we've got the similarities in our genetics, so we can look at their genetics and learn a lot about ourselves by looking at how the genetics may correlate with the behavior of the monkeys, as well as looking at environmental effects and how that might play a role. So if you'll go on to the next slide, this is just a little bit of a summary of a paper that was published recently by some of our collaborators. We looked at the effects of a natural disaster on the aging of these monkeys. So it was an unfortunate experiment that was not planned, but because these have been studied for so long, that when the island of Puerto Rico was hit by Hurricane Maria, Cayo Santiago was right at the front of that where it was hit first. And so you can see by the before and after pictures here that it was a lot of the vegetation was destroyed. And so thankfully only a few of the animals were injured from that. They apparently know how to hunker down well. But obviously it's made a drastic change to their environment, and also the stress of the event can also have an effect. So our collaborators, they looked at the gene expression in the blood of the monkeys, and they have this data from before this event and then collected some afterwards, looking at their behavior and their genetics. And by looking at how the gene expression changed, and these are particularly in the immune cells because that's what cells are in our blood, they were able to show that these animals that experienced this hurricane actually aged more biologically than so basically it added about 2 years to their age, which is equivalent to about 7 to 8 in human years. And so we talk about biological age is like basically our body's age versus our chronological age of, from birth. So some things can, cause our bodies to age more or less. And so this is something that was shown to show an increase in aging due to the stress of this event. And with the continued study of these, perhaps they'll be able to see more of how the long-term effects are and potentially resiliency after. So this is just a quick snapshot to show you some of the type of research that's coming out of these broader studies of these monkeys. So if you go on to the next slide, now I'm going to focus on our specific project. So Monkey Health Explorer is particularly focused on looking at the blood. And so our main research goal of our project is to examine whole blood from these rhesus macaques to help us understand health and behavior. So connect the health to the other observations. And so the work is done, like I said, just like when we go to the doctor and get our blood drawn, the blood is taken, a sample taken, it's smeared across a microscope slide and stained so we can see the cells more clearly under the microscope, like the picture here. And if you go on to the next slide, so this is a little bit here of like what is in our blood. So here, the little cartoony thing here is just to give a sense of the percentage of what's actually in the blood. And the picture above is an example from our monkey slides, the blood smears. So you see that our blood is more than half of it is plasma. That's the liquid part. It's mostly water that contains the cells and some proteins. We got 40%, 42% of the blood is red blood cells. So if you look at the picture at the top, it's those little discs. There are a lot of them spread around. Sometimes they look like little donuts, and that's because of the shape of red blood cells. They have this shape that causes them to go in the middle. I like to say it's like a donut that's been squished, like a jelly donut that's been squished in the middle. And then, of course, red blood cells are important for moving oxygen around our body. Then we have 3% of the cells are platelets. Those are the ones that help us clot when we get a cut or scrape. And then just 1% of the blood is made up of the white blood cells. And that's the ones that we're focusing on in our project, because those are part of our immune system. So, you can see in the picture above two of the white blood cells that are pointed to there. But go ahead and move on to the next slide. So here are some example pictures, and this is one of our other goals of our project. So in addition to gaining the research information to help us in studying these monkeys, we also want to give back to our volunteers by offering them training. So we want to train people in identifying the types of white blood cells. They're gaining a skill by participating in our project. Plus, this actually helps reach out. We've had students from like vet schools and med schools, as well as trained technicians that's their job. They like to do our project as a practice or just to use the skills that they've learned. So that's a lot of fun that we get the feedback from some of our volunteers. But here we show images of the 5 main types of white blood cells that we're looking for in our project. And the white blood cells, the bigger one in the middle, and then there's little information about what each one does. And you see how each one plays a different role in protecting our bodies. So neutrophils, they're first to respond to bacteria or virus. We've got lymphocytes that create those antibodies. Monocytes are the ones that go around and clean up dead cells as well as killing invading cells. Eosinophils are involved in allergic reactions, as well as fighting parasites. And then basophils are also known to be associated with asthma. So we've got these 5 different types. And in our project, we train our volunteers how to look for them, how to identify them by the patterns in them, in order to help us gain that information about the health of these monkeys. And by looking at how many of each type we have in the blood, that's how you tell if an individual is, within normal ranges, or if one of them is perhaps elevated, like say an elevated amount of eosinophils might indicate that the person either is having an allergic response or possibly have a parasitic infection. So those numbers can tell us a lot. So go on to the next.
I'm going to ask a quick selfish question.
Yes.
I'm asthmatic, so I'm curious, is having a lot of basophils is more likely that you have asthma, or what's the...
It's a response to foreign invaders, basically. So it's triggering, but I honestly, again, I'm a geneticist. I am not trained in, so it is a great question though. I only know it's part of the process of in an asthma response that I believe they're triggered, but it's not that they're causing it.
Okay.
They're a response.
Gotcha.
That's my understanding.
Presence of them might show asthma.
Indicate, yes, that there's been a response. Yes.
Excellent. Okay, thank you. Sorry.
Yes, no, great question. So here's a question just to see what, people think. Which do you think is the most common type of white blood cell in humans? So we've got the 5 types here, and I like to say them because some people are like, how do you say that? So there's basophils, eosinophils, lymphocytes, monocytes, and neutrophils.
I know from my minor experience with blood cell knowledge, lymphocyte is the only one I recognize but it might just be because of lymph nodes, lymphatic system, everything.
Right.
So I don't know if that would be, I don't know if it's right, but that's the one that I would use my gut answer. And according to the poll, I'm already not correct, or at least someone else thinks they know better. We'll wait another couple seconds.
I will say that one of our viewers knows quite a lot about the project. That would be Dr. Julie Horvath, who is the head of this project in our lab.
Oh no, you cheater.
It's a little bit of a cheat there, I will say.
Well, it looks like most people are scared to answer this question, to be honest. So I think our answer is coming from the project leader themselves.
Probably,
that's my guess.
yes. So you can move on to the next slide, and yes, it is neutrophils. Go ahead, there you go. And they're the first responders, so it makes sense that they'd be the ones that we have the most of. You can go on to the next slide. So as far as like who participates in our project, so and our goal is to have our volunteers learn how to identify the white blood cells and contribute to our research. But it's been neat. The ones that have interacted with us, we have students that are using these basically using this project in classroom, like their teachers have brought it into the classroom. We have, like I mentioned, veterinary students, medical students who like to use it as practice for identifying blood smears, as well as trained technicians who they think it's fun to use the skills they do in their job in this different way. So, but we also have retired individuals who are just interested in science and participate in everyone of all different range, basically.
Uh-oh. Sorry, my screen share decided to just drop off. That was rude. Here.
I'm gonna get some water real quick.
Good. I did that on purpose then. Needed a water break. All right, there you go. Sorry about that.
Okay. And then so far, like I mentioned before, we have more than 15,000 public participants, and that's actually just the ones that have registered for Zooniverse. We do have unlogged users, which adds, I think, at least 9,000 more people that we have, at least 9,000 sessions. Excuse me. And so we have images, like blood smear slides from at least 900 animals. And so far, since we had this project going, and it's been going for 5 years, I believe now, we have gone through almost 1 million blood smear image classifications. That means like individuals analyzing the cells in an image. So we've got a lot going on, and basically it's for anyone who's interested, particularly if you like pattern matching, like games where you're looking for things like the lost, hidden objects. It's kind of like that. So go ahead on to the next slide. So how do you participate? So hopefully I've intrigued you enough that you might want to check us out. So you can get to us if you go, I do highly recommend you go through SciStarter because it makes it easier to connect and keep track of what you've done, but you'll learn a little bit more about that. But for now, go ahead to the next one because what I'm going to do now is give you a little bit of a starter of a tutorial of how to identify these cells. Because if you go on our project and you start in the beginner project, there's little tutorials and there's a lot of other information to help people identify the cells and figure out what they're looking for. But here's basically how I like to do this one. Sometimes I bring this into classrooms. I did failed to mention before that we do have educational materials on our Zooniverse page. We have curriculum correlated materials, and I've been able to take this to a couple of middle school classrooms through the SciMatch program a couple years, which has been a lot of fun when I've been able to bring this and some fun things like 3D printed rhesus macaque skulls and candy blood smears. Those are some of the things we use. Use different candies to represent the different types of blood cells. So those are some of the fun things that are described in those materials. So anyway, so we go through and explain what patterns to be looking for to identify these white blood cells. So we've got neutrophils and eosinophils. I'm showing these at the same time because they're kind of similar, and so they can be easily confused for each other. So I'd like to show them at the same time. I have under them, again, what their roles are, just to give a sense of, see how they each have a different role. Now, the first thing to notice is that the white blood cells are the big cells in the middle of the picture. So they're like 2 to 3 times as big as a red blood cell. So that's one thing that makes them stand out, makes them easier to identify. Plus, they have the dark shape on the inside. So, and can you guess what that dark shape is? Do you know, Emma?
Oh gosh. Embarrassingly, I used to like teach 5th grade science.
It's the thing that contains the DNA.
Yeah, the nucleus.
Yes, it's the nucleus. and you know, the reason we don't see that same kind of dark shapes in the red blood cells is because when they get mature, they actually lose their nucleus. So in the white blood cells, they really stand out because you've got that darkly stained nucleus. And that's the shape, that's what you really want to pay attention to. You'll notice in the neutrophil that you've got like 4 or 5 blobs, kind of small, but I like to think of them as like beads on a string. They're connected together, but those are the different lobes of the nucleus. In the eosinophil, there's really 2 kind of bigger blobs that are connected. They look almost like a sideways pair of lungs, or I don't know, think of other things. Sometimes they look like a pair of glasses. And that's one of the big differences between them. Neutrophils usually have 3 or more of the little blobs, and eosinophils usually only have 2 or occasionally 3. So the number of shapes, that's one thing to look for. But another big difference is the background. So the cytoplasm, the rest of the cell, you'll notice it's light in the neutrophil and it's darker in the eosinophil. So basically the difference is because the eosinophil has little granules in the cytoplasm that give it a kind of a shaded look. I like to describe it as kind of like having sand in there that's just giving a little bit of darkness. And you know, looking at these, you can really see the difference in these example pictures. But unfortunately, from slide to slide, some slides are brighter than others. Sometimes the stain takes better than others. So you can't just look for a specific shade of pink, and that's a neutrophil specific shade. So you want to look at the relative brightness. And I like to tell people, in order to figure this out, look at a nearby red blood cell, particularly the ones that are like the donut shaped. So look at the top right of a neutrophil exactly. Inside the donut hole looks about as light as the background of the neutrophil. So see how that's very similar? And then if you look at the eosinophil, there's several donuts above it, of the red blood cells, and say how the outer ring of the donut is more like the background of the eosinophil. So see how you got the darker part is closer to the eosinophil background, and the lighter part's closer to the neutrophil background. So that's one of the things to look at. So even if you have like a really bright picture or a really dark picture, looking for the red blood cells to see what that difference is, that's how to help and look for that background. So between the 2 of those, you should be able to figure out, as far as looking at the number of shapes and the background, figure out whether this is a neutrophil or an eosinophil. And then a little bit of other information is that neutrophils are, again, most common. We mentioned that 40 to 60% of the white blood cells in humans, and we're using human numbers because we're still developing the standard for our monkeys, and so it's still they seem to fall in the same ranges that humans do in general from what we've looked at, but we're using our human numbers that are well established as our standards for the moment. So all the numbers I show are from humans. So 40 to 60% of our white blood cells are neutrophils, only 1 to 4% are eosinophils. So let's go on to the next slide and maybe see if we give you a chance to try out, see if you've learned something. So which one of these, A, B, C, or D, is an eosinophil?
Ah, I have my answer, but I can't answer. Or do I? No, I do. I was worried, but I'm really glad you added these to the side.
Yes, I added this because that's in our project. We have all sorts of examples that you can use to look at while you're looking at the slides. So you're not having to go from your memory. You can look at pictures and try and see which ones look the closest. So look at the numbers, look at the background. See, do we have any takers?
I do agree with what has been chosen in the
Yes, that is correct. So B is an eosinophil. Yay, because it's got that darker background. It just has 3 shapes. And then move on to the next slide, because the next question is which ones are neutrophils?
Oh, could there be more than one answer?
There could, which I don't know if your poll can do more than one, but that's okay. People can guess what they think. Oh, it is multiple choice. Very good.
Oh, that might also just mean that people have to choose one though.
Oh, but right.
Oh
It's
wait,
okay.
never mind. I think we're okay. Or not being able to see it. I agree with that person.
Okay.
So I feel good about my answer now. It's
And
nice
this is a good
side.
example picture because it's got the nice, easy to see lightness.
I was gonna say sometimes like I love the Project Sounds universe and I look at like the early on ones and then you get to the real project, you're like, this is hard.
Right. Yes, yes.
I was tricked. No, but in a fun way, but it's still like
Exactly. Yes.
oh, man.
So yes, A and C are the neutrophils. And go on to show. So the question then is, last question, what's D? So move on. Is it a white blood cell? Is it a red blood cell? Is it a platelet? Is it a parasite? And I mentioned parasite because that's one of the other fun things that are some of our volunteers, the ones that are either students or professionals, they have sometimes given us diagnoses or just their thoughts about the blood smear, like, oh, I see what looks like a parasite in this, or maybe they'll see something. There's something called basophilic stippling, which puts little dots inside the red blood cells, makes them look a little bit like chocolate chip cookies. It's not a good thing because that could indicate either heavy metal poisoning or like a parasite. But we've gained a lot of extra information from volunteers that are pointing out things like that in our pictures. So it's been a cool add-on. Yes, it's a red blood cell. Good job.
Oh, okay. I swear that's what I was going to say too. It was between that and platelet, but the little guys are platelets, right?
Yeah, the platelets are little bitty dark spots.
Yeah.
So
I was
okay.
curious if since it's like a staining technique, does that mean anything that might be a parasite would still be the same color? Does it just look like what would it be shaped like?
Well, it depends on the type of parasite. Sometimes they'll often, if they're in the early stage, there'll be tiny circles inside the cells. So they are you have to really know what you're looking for. Again, I'm slowly learning some of these. I don't know that as well. But there have been some that have actually been outside the cell and they're like worms looking.
Ooh, yeah.
So that's a lot less common in here, but they do, because it's staining certain proteins, they often will pick up some of that stain as well.
Okay. Fascinating.
Yeah. So you can keep on, yes. Now I'm just going to go through the other ones really quickly. So here we've got the other pair that I like to talk about together, monocyte, lymphocyte. Monocytes are also as big as the other two we talked about 2 to 3 times the size of the red blood cells. But lymphocytes are the only white blood cell that's not usually that big. It's usually only a little bit bigger than a red blood cell. So that's a big key in looking. If you see something that's got that nice dark, nucleus, but it's just a little bigger than a red blood cell, then it really makes you think it might be a lymphocyte. The other thing to look at is this pair. I like to put them together because they both mainly have one big dark shape in the middle versus the other one. The previous ones had several. Now, technically, the monocyte actually has 2 lobes, so 2 blobs, but they're smushed together. So often it looks kind of like a ball of Play-Doh that's been poked into. So it's not smooth. Whereas like the lymphocyte is where you like take that ball and you roll it around so you get it mostly round. So that's the big thing, is the shape. It's usually irregular in the monocyte. Often it's kind of C-shaped, and the lymphocyte is mostly round. Plus, you don't see a lot of cytoplasm around it. There's not a lot of that clear space around it. you see more of that in a monocyte. They both have, though, the lighter background, so they don't have the darkness that the eosinophils had. And then the last thing to look at, of course, is thinking of how common they are. So the lymphocyte is the second most common of our white blood cells. So when you were thinking that earlier, you were close. Very good job. And so they're 20 to 40% in humans and of the white blood cells, and then monocytes only 2 to 8%. So a lot less common. So again, it's like with the question of, if you're in doubt between choosing between the two of them, go with the one that's more common, because you're more likely to be right.
So many lessons in this.
Yep.
I can see why it was developed for educational tools too.
Yeah. And it's interesting, the more you look at these, the faster it is for you to quickly identify, because you know, if you've done it enough, your brain really picks up on the pattern matching, which is one of the things the brain's really good at. So go ahead on to the next slide and we'll show you the last one. So this is a basophil. Basophils are, they are, again, 2 to 3 times the size of a red blood cell, but it does look very different because it's like all dark. And it's because this has really big granules, bigger than the ones in the eosinophils, that really take up a lot of the space. So the nucleus on this one is on the left side, where it's kind of a lighter pinkish purple, and then it's darker pink purple on the rest of it. So the nucleus is often kind of in a little bit of an S shape or sometimes a C shape, but it's almost completely covered by all these dark granules. So they stand out because they're really dark compared to the rest of the white blood cells. And so they're also extremely rare, less than 1% of the white blood cells, because they're typically only there when they're responding to something. So there's just a few that kind of are around during normal times, basically. So this is one that, you know, a lot of people get excited when they're like, oh, I think I found a basophil, which it is kind of fun to find something rare, but we also don't want people to just think everything's a basophil because they want to have the rare thing. So, but it is interesting. We've definitely don't see it for a while, and then you'll see several, and so it's kind of interesting. But that's the last one of the 5 types. Go ahead on to the next. We have one more question to see, just to not to do all of them. We're just going to do B. So which type of cell do you think B is?
Ooh, I have my thoughts.
And I have the pictures of all 5 now, so you can really see them all.
For the cytoplasm color, the shape of the nucleus, right?
Yep, nucleus. Yep, you're correct.
And I think I'm right, because whoever's in the poll is definitely making me feel.
Yeah. Let's see. I'm guessing it's going to be right. Yes, it is a lymphocyte. So do you have a guess even though it's not in the poll, what is A?
Ooh, oh boy. I feel like A and D are the same.
Mm-hmm.
And they would be, oh yes, I think it'll be the neutrophil.
Yep.
Maybe.
How
And
about
then
C?
C looks like a eosinophil.
Good job. You are correct.
It's
See,
also
you're learning.
Big. Yeah, I feel very smart right now. Thank you. It does look massive though. Am I just like, it looks like it could be confused with several different cells? Is it really just one?
That's just to show how it does vary a lot. And that's one of the things that comes up. A lot of people ask us, well, why don't you just get a computer to do this? You know, machine learning, AI should be able to. Well, it's actually really hard because there is so much variation. So far, we've got several groups we've worked with, and we've got some progress. But often they've done good at particularly identifying what is a white blood cell out of the picture, but not quite in identifying the types. A lot of that just because of the variation in the staining. Our slides are made in the field in like a little trailer on the island, so it's kind of rough conditions, not like a nice, you know, clinical lab where everything's all clean and you air conditioned and all that. So it's a little rougher. So I think that's part of it as well. So.
Yeah. If I had a quarter for every time I have said the commentary about AI is just not there yet, like our machine learning just isn't there yet, I probably wouldn't be a millionaire, but I think I'd have quite a bit.
Right. A bit. It's definitely promising though. So
definitely, yeah. It just takes time. The more people get involved now means that the machine will learn better and better over time. So even if you just care about computer science, this is a great use of time. I'm gonna pause on the slide here just for a moment here because the reason this is out of here is because since it's through Zooniverse, it's actually a SciStarter affiliate and you can receive credit and you get to track your contributions over time. So if you're interested in joining the project, like I definitely am, I would be going onto my SciStarter account, going onto the scistarter.org/monkey health explorer, clicking visit to be redirected to Zooniverse. And on Zooniverse, I already have an account, but if you don't, you'd make an account and take note of that username. And then my username, which does already exist, is actually put into the affiliation integration, which is in your account settings, which for anyone who is unsure, I'm just going to go ahead and click that to show you what it looks like. So there's me looking like a nerd. Love it. Caterpillar Research back in the day. And then down here, affiliation integration, I would write my iNaturalist username is there too, but you add in your Zooniverse name and then you're all set to receive credit. And then you go back to the actual practice. So on the Monkey Health Explorer project on Zooniverse, you can do beginner's practice, start here. And then I'm gonna go ahead and drop my screen share for a moment and hand it over to you so we can see what that actually looks like. And if you wanted those links, those are in the chat as well. Roland dropped those for you. Hopefully you're able to screen share. Let me know if you're having issues too. Perfect. Oh, I'm sorry, you're muted. That might be why. I was like, what's happening? Just talking to myself.
There we go. Found the button. Sorry. That was me being slow. And let me, can you still see, you see the Zooniverse page, Monkey Health Explorer? Okay, just checking. I had to move it to the window I'm looking at. So if you decide to check out our page, go to our page, you can, Monkey Health Explorer, you can learn more about the project by clicking the about and reading all about the research and some other information, as well as the educational materials are there. But when you're getting started, you go, we always suggest you go to the beginner practice. And when you first go on there, the tutorial pops up and it starts going through a lot of what we've already gone through today, which is just showing the basics about the different cell types. It also does show how to use the tools in Zooniverse, so you know how to move around the picture, how to mark it. There's also what's called a Goldilocks test. This is in a lot of our workflows where we are asking how well are the cells spread in the piece of the picture you're looking at, because we really want the ones that are just in the good part that are spread nice and evenly, but we still will take information from all of it. And so we just want to know, is it too sparse, is it too thick, or is it just right? So that's why we call it Goldilocks. And then after that, you learn how to, when you click on a white blood cell, it'll give you this drop-down list to choose what type it is. And then there's also a video tutorial that talks through, it's me talking through what we just went through of explaining what to look for inside the different cells. And then it says, you know, a little bit more about talk. I'll show a little bit about that here. But I do want to point out that, you know, one of the great things, if you haven't been on Zooniverse, is you've got this field guide option on the right with lots of information. So we've got more video tutorials. We had a wonderful volunteer who created a bunch of extra tutorials for us that you can see on here, which is marking cells, using the talk boards, and specifically identifying the white blood cells. But we also have, you know, FAQ. So white blood cell examples. This is something you could pull up on the screen next while you're working, so you have extra pictures. We even have a printable version, a PDF file, in case you really get into it and want to print it and have it next to your computer instead of having to keep looking on the screen, to have lots of example pictures. But in the project, once you get going, so here is our beginner practice. And it tells you here, you know, what you want to do. You want to look for white blood cells. So you look for these bigger cells. And in the tools up here, we've got the plus sign will let you zoom in. The big arrow plus lets you, it's the pan, allows you to click and drag the image around so I can move those cells where I can see them. And the arrow allows you to actually select a cell. And I'm going to choose that. That's an eosinophil. And here is a neutrophil. So it's that easy just to choose which kind you think it is. And then, okay, well, I know there was some more on this image, but now I can't see it. There's a couple ways you can get around. You can either click on the pan tool again to move over to wherever, see if you can find the other cell, or you can click the bottom button that takes you back to the zoom out. So that's just a little bit about how to get around. And then I will go ahead and mark this last one over here, which is also eosinophil.
one of the tips I got from the marine iguanas or iguanas from above project on Zooniverse was they have a scanning technique they recommend from going zigzagging
Mhm.
across. And
Yep.
that changed the game for Zooniverse for me because I've always had trouble picking out the details. And so
Right.
anyone who's listening that might be benefited from that, definitely
That
use the zigzag method.
Yep, that's excellent. That's what we tell when we have our volunteers, especially when we started out looking on actual microscopes, looking at these images, and you start in the left and look across, and then you go back and you just zigzag across the whole screen till you get to all of it. So excellent suggestion. So for the practice here, once you're done marking, you click next, and then it tells you, oh, well, you can look at at the answers to see if you're right by clicking at the bottom of the screen there. You see there's a dark circle and a white one. You click on the white one to look at the next picture, and it shows you the answers. So this is our way to let people give it a try and see the answers and see, okay, and you can even on this part of it, you can zoom in again so you can look more closely and see, oh, like if you got one wrong, you can look and see what you should be looking for, see why you may have got it wrong. And then when you're done, you can either click done or you can click done and talk if you want to maybe make a comment. So here's for this image where we've had several people who commented about the project, and that's just about the specific image basically. So that's one of the talk options in the project. So if you do that, you can just go back to the main page and go back to your workflow. Once you've done enough of the beginner practice, you feel like you're ready to go, you can then choose one of our other workflows. We have our standard workflow that's identify white blood cells. We're on set 7 right now. And that's showing the same kind of images. We also have one that we're testing a new method where we're just showing single cells, which again, the tutorial always pops up the first time you go somewhere. But here it's showing, and this is actually was using AI to just identify white blood cells and then cut a version, like the space around it. And we're using that as a test to see if this would be a better way of doing our project. So we ask what's in the middle. Well, this one actually looks like it's a cell that's fallen apart because it's kind of, if you notice, there's no real cell membrane. And again, you can zoom in, but it's like a little bitty picture. So this one, there is an option of no white blood cell debris because sometimes it's identifying what used to be a cell, but we don't count them anymore if they're no longer intact. So this one we would choose no white blood cell. So this is another workflow options. If you want to do the big picture, you do the standard workflow. The single cell test is another one. We're almost done with this particular test, and then we'll decide if we want to which way we want to go. We may keep both. So those are the different options. You can also, if you want to get more involved in talking, you can talk with the researchers about the research in the talk option. There's even tech support if you have some questions, and then chat as well for just general chat with other volunteers. And that's where there's information, even how to track your volunteer time, classifications, if you're not already doing that through SciStarter, which I still recommend is the best way to do it. So that's the quick overview. So do you have any thoughts, Emma, on things that you think would be a question if you were doing this?
Yeah, I'm trying to, I mean, I've never felt more prepared because of the tutorial we just went through. So I mean, if I'm doing this on especially since funny enough, the identify white blood cells, like singular one single cell test where it's the burst one, I was going to ask about that, but reading those options, I'm glad that that's noted as a part of it. Yeah, I mean, the zigzag method and I feel way more confident to do this one than I have in a lot of them. So
Well, great. I'm glad to hear that. I
yeah.
will say one thing. I will say that sometimes if you're in the mode where you're marking cells and you click, which something is not, oh, got a pop up on my computer. Huh. Well, that's a tech support issue. I am zooming, but it's not letting me click. I'll have to check that out. Let me go to a different one. So, oh, it might be because I'm not signed in. I don't know. It's no longer letting me click on objects.
Oh,
let me see if I do it. I'll do it on here. So, one thing I wanted to point out is if you were doing this project and you were like, oh, I want to zoom in, I want to move this around, but you were accidentally in the arrow mode and you click over here. I'm like, oops, I don't want to click. I didn't want to mark that. You can just click the little X and get rid of it. Or if you accidentally click, but you're like, oh, I really want it to be over here, you can move them around. I just like to point that out because sometimes you can accidentally have a mark. You're like, what do I do? It's not a cell. It's not a white blood cell. So that's one thing I did want to mention.
Definitely. It's nice that it's or I'm curious actually, in one of your other protocols that are listed here was count red blood cells. Is that
Mhm.
that would mean for every single one, I assume, right? Every
Yes,
single
those are our Zen projects where that's what I like to say. If you like to be just very Zen and just click, 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 you're not having to count yourself, but you're just clicking on everyone. So this is the platelet one where you literally would just click on every platelet you see, and
Mhm.
then on red blood cell you'd click on the red blood cells. Which it is. We have some users who really enjoy doing it. They say it's almost like meditation.
Mhm.
And this is something to feed the pipeline of sending it through with having the computer doing the counting, and we can compare the numbers. So that's why we have those set up. And that's why, we have them over here as just, we marked them as tests, is that you're welcome to try these if you just want to just mark cells. Um, it all contributes to our project in whichever way you wish to participate. So, um,
That's excellent. I'm wondering if, in reference to people going through SciStarter and participating, hopefully in April too, which I'll talk about in a moment here. But I'm curious if you had a million people, or not a million people necessarily, but 1,000 people who contributed near a million entries or contributions to the project. I'm curious what the immediate results would be. Like how much out or how much research can we complete? I guess I'm not sure what the right question to ask here is, of like what we would know after doing that much.
right. Well, I can show you here a little bit of this. These are some of our workflows, the ones that have completed, where
Mhm.
you can see how many images were processed.
Yeah.
And so these just show like basically how many classifications. So we have at least 15 people mark every image.
Mhm.
So that way we can, it's the whole thing of consensus. Like if you have enough people look at something, then the consensus will be the right answer. So it takes at least 15 to look at every image. And like this particular data set is almost 6,000 images. So if we had that many, we could like possibly complete the current web sets, and we'd be able to then upload more images. We do them in sets by ones that are the same set of animals collected at the same time. That way we
Mm.
can have them in data sets. And so like we have slides ready to go. So if we had a big bunch, we could just like zip through these. It would give us a lot more information, and we could start providing summary information about like what we see, because this is where we look at the counts of like the percentages of the different types of white blood cells. We can, if we have enough of these, we can start having what is the standard range for these rhesus macaques, ones particularly on this island, this population, by having enough individuals measured, basically. So it gives us the baseline to then see which ones are out of that baseline, which ones, have an elevated amount of white blood cells of a certain type, and then we can follow up with the vets to ask if they have seen something when they've been working with the animals. So that's some of the information, what it can go to in like the short term. And then it of course can be connected with some of the other researchers with their work where they're looking at other aspects from the same animals. So it just gives us more to work with, more data to dig through and connect up.
Yeah, absolutely. Being able to see like, oh, this is out of the standard. Do you see differences between this rhesus to the next one? Or what makes them odd? Is it
Right.
behavior? Is it some kind of health something? Any kind
Right.
of knowledge? Yeah, that's
Yeah.
excellent. Yeah, it would be really cool to get all those in April. In
Yeah. Yeah.
yeah.
Yeah,
Um,
like as far as thinking about like how does this relate to behavior, one thing I like to say is, I don't know about you, but like when I'm sick, I'm not exactly the most friendly person. Like I'm not really sociable. And so I can imagine that if they're seeing an animal that's behaving that way, by looking at their health, at least at the time, like, okay, well, maybe this explains why the animal was behaving that way. Or if they were perfect, everything looks good, then it must be, something else is going on. So it's one piece of the puzzle.
Oh yeah, that's very true. The older you get and the more you know, it's the bigger puzzle. It gets to be a bigger puzzle.
Yes.
It's excellent. That's awesome. I mean, I not necessarily about the actual project, but I am curious now that I kind of steered the conversation towards future events. We had a conversation before this about activities during April and Citizen Science Month. So I wanted to give you a chance to share if what your plans are to encourage people to join between now and also just during April when we have such a big push to get 1 million acts of science and get as many people involved as possible.
Right. So yeah, we are definitely signed up to participate in the 1 Million Acts of Science in April. And the one thing I know we're definitely going to be doing is here at the museum, we have an event in April called SciTech Expo, and it's going to be on April 27th. And we'll have all sorts of cool things set up throughout the museum, tables with different kind of activities and information. And I'll be sharing about this project then. It also happens to be the grand opening of a new exhibit in our museum that's hopefully will be bringing lots of people. So it gives us more people to talk to. But I'm also working on plans. We're thinking about doing some sort of a hackathon earlier in the month, or maybe a couple times in the month, where we'll be actively on our Zooniverse page, like really there to talk with people in the talk, to get involved, so that maybe we can get more people involved by getting a chance to actually talk with us more one-on-one.
Yeah, I'm really excited for that. If when that comes out, or if things are decided there, I'll be online if I can. Working on it too. Especially
Great.
since I mean, I'll do it before then too, now that I know how to do all of it. That honestly, best tutorial ever. And we have a question in the chat about what your favorite type of monkey is. Piggybacking off of that, actually, I am curious how you got involved specifically on the or like why monkeys in this case, since it wasn't your original
Right.
path. But yeah, favorite monkey?
That's a tough one, because there's like I I yeah, I should know to have an answer to that, but I don't really have. I have a specific one in my mind. I mean, I do particularly like those rhesus macaques, because I get to I see the pictures of them all the time. So like they're kind of my go-to monkey right now, I guess I would say. But as far as how I got involved in the project, so in our lab, so Dr. Julie Horvath is the head of our lab, and she's the one that has the collaborations with our other team members there. So I just got connected, and by I'm the lab manager here in our lab, so being connected with her basically introduced me to the project and got me involved. And it's been fascinating. I've loved it, and it's become a much bigger project than we were thinking as far as creating the Zooniverse page. And it's been so much fun interacting with our volunteers. And I did want to mention, we've had several people step up and translate our project into multiple languages. So we didn't even know that was an option until someone initially stepped up and asked if we wanted it in French. So it's currently in French and Arabic and Chinese and German as well. And we have a couple more languages that are underway. So that was just a fun, and that came completely from our volunteers.
Yeah, dedicated people out there who just want to help. That's too funny. It's funny because Roland actually is trilingual and has been translating projects too for Arabic and French. And so it's fun to hear that there are other people around doing similar things, because I mean, it's worth spreading the love across other countries, right? Making sure it's accessible to all. So I'm glad that's already in works. I was like, oh, if you don't have Arabic, Roland's got your back.
Yeah, we've got Indonesian, Russian, and Italian, and Portuguese and Spanish are
I was
the
going to
ones
ask
that are
about Spanish.
yeah. So I mean, yeah, it's great.
Yeah, that's a lot of languages. That's awesome. I'm glad that's like way more than I've heard of for a lot of projects. I mean, I'm glad that people are so excited about this one. Makes a big difference, I hope, for those who are like, I want to be a part of this, but I don't know how
Right.
without the language. Not that anyone asked, but my favorite monkey right now is the bonobo, because they're a matriarchy, and we're about to hit Women's History Month, and so even though they have other weird things around their species, they're a matriarchy too.
Right.
Excellent. So we're nearing our last 5 minutes, so I'm gonna start closing us out. Thank you so much for explaining all of that and for the excellent presentation and interaction. I have never felt smarter and more prepared to do a project, as I mentioned before. So it's very, very
Thank you. It's been great being here.
awesome. And the last thing I wanted to mention was for anyone who's joining us online and on Facebook as well, if you're curious about NASA projects, both for space science and also for earth science, we have an event next week that's taking place in lieu of our SciStarter Live. It's a Do NASA Science Live. So if you join us next week on the 14th, or on the 14th, that's tomorrow, on the 21st, you can learn about icy comet trails and snow on Earth and all kinds of frozen landscapes. So we'd love to have you join us for that. It's at 7 p.m. ET, and that's what's happening next week. And Lastly, if your interest was piqued towards anything about April earlier, you can sign up as an individual and as a project leader, like Marianne already has for the Monkey Health Explorer by going to 1millionaccessscience.org or citizensciencemonth.org. There's a bunch of different pathways. But if you go to the Science Starter website, you can get there and start participating. I believe Roland also dropped the link in the chat, which is perfect. Thank you for that. And it just means that we're gonna just go crazy during April, getting as much citizen science done as humanly possible. So help out your favorite projects, connect with your project leaders. They all are excited to share the project with you and just push the research forward within 1 month of excellent help. So if you need any assistance with any of these projects or connection with us or help with using these things in your classrooms or connecting with the project leaders, we want to support you. We are an educational hub for a reason, SciStarter is. And so you can ask your communities, you can ask the project leaders, you can email us at info@scistarter.org. You can find more projects and save them to your dashboard if you have an account. We have a podcast that explains a lot of these projects and talks about the news in citizen science. So always a good thing to be a part of. And also the training if you want to get those LinkedIn badges that I love so much. But otherwise, I mean, we are excited to see you hopefully next week for Do NASA Science Live. And thank you so much, Marianne, for joining us for Monkey Health Explorer. This has been really excellent. If you feel that the desire to take our survey, it's literally will take you less than 2 minutes to take. You're welcome to do that too. I think I've done it in like 30 seconds as a practice round. So I know it can be even shorter. So feel free to share with us your knowledge of how this went and let us know if there's anything you want to hear about next, and we'll add it into our calendar. Thanks everyone for joining, and we'll see you next week. Thanks, Marianne.
Thanks. Bye.